Good morning, class. I hope everyone's having a great day when they start watching our video. Today we're going to be learning about meiosis. So we've started with mitosis, but now we're going to get into meiosis and learn about the differences, and luckily there's a lot of similarities. So our first question is, what acronym do we use to remember the stages of mitosis? So if you guys could remember that one from earlier this week or a little bit in last week, mitosis, we use our acronym IPMAT-C. That's how I like to say it. Sometimes IPMAT is just enough, but these are our acronyms for each stages. So of course, using this acronym, what are our stages? Well, we have interphase, which is I, prophase is P, M is metaphase, A is anaphase, T is telophase, and C is cytokinesis. And then we have another question is, why do siblings, which come from the same parents, look differently? So if we were to have mitosis, we say that we have identical cells, daughter cells. The products are identical cells. But when we have meiosis, we don't have identical sisters or identical brothers. So why is that? Well, it's because meiosis creates genetic variation. So we'll be learning about that today. So our objectives for today is to understand the basics of meiosis and then list and describe the steps of meiosis as well. So we have a great video here. Please pause this and go watch this. It does a good job explaining it. And then to get into our basics. So our basic definition, meiosis is the making of new cells through sexual reproduction. So this also causes genetic diversity, and we create 4 different cells. Well, they are called either egg or sperm cells, and they each have 23 chromosomes. So if we have a female, whether mammal or even different plants do this as well, lots of different eukaryotic cells do meiosis. If the female part creates an egg, and then the male part creates a sperm. So we get 4 different eggs out of our original cell that goes through this type of division. And they each end up with 23 chromosomes each. And then we have the same steps as meiosis. We still use our IPMAT, but we just do it twice. So we have our picture here. We have our starting our interphase, and we have our chromosomes start to show up. We have them start to get pulled apart or line up in the middle. This is the middle. So this is our interphase. This is our prophase. They start to see our chromosomes. Metaphase is they line up in the middle. Then they start to get pulled apart. This is kind of like anaphase and telophase together. And then we end, after meiosis 1, we end with 2 cells. And then we go through the same steps again, prophase, metaphase, anaphase, telophase, and end up with 4 total cells. So it's just like doing mitosis, but twice. This is another good picture of it. So we start in interphase. We go through prophase 1, metaphase where we line up in the middle, anaphase where we're pulling away, so away, anaphase, and then telophase where we start to separate, and we see these new nuclear envelopes being made. Then I would say we also have a cytokinesis one as well. Then we have prophase 2, so we have 2 new cells now, and we're going to go through our same steps, but with each cell that was just made from meiosis 1. So meiosis 2, we have prophase 2, metaphase 2 where they line up in the middle, anaphase 2 where they get pulled away, anaphase away, telophase where we start to see nuclear envelopes and they start to pull apart, and then finally cytokinesis 2 where we have 4 new cells. So here is an idea of how we would count how many chromatids versus chromosomes there are. So chromosomes are, for example, we have 23 chromosomes in each one of these two guys, so this is the idea, and then we still have 23 chromosomes just by the way they're labeled. I think what makes more sense is to count our chromatids. So this picture shows us that before interphase, we have 46 chromatids in one normal cell. But after interphase, we have gone through DNA replication. So after we replicate every one of our 46 chromatids, now we have 92. 46 times 2 is 92. Great. So then after our first meiosis, or meiosis 1, where we end up with 2 cells, we go back to each of them having 46, but then we do another division where we end up with each of them having 23 chromatids. So we start with 92 up here in the very top, then we do 92 divided by 2 because we have 2 cells in the after meiosis 1, and then 92 divided by 4 ends up being 23 because we have 4 new cells all with 23 chromatids or 23 chromosomes. So we're going to kind of step through each stage like we did for meiosis, but we're going to go a little bit faster because we've already learned about it. So interphase is the exact same as it was in mitosis. We have our G0, our G1, our S, and our G2 phase. We have our DNA replication is the most important part of our interphase. So when we think interphase, think our DNA is replicated there. And this ends up leaving us with 2 copies of DNA. And we also have some centrioles move away from our nucleus and prepare for cellular division. Our prophase I is just like normal prophase, but we have one interesting thing that happens. We still have our chromatin shrink to chromosomes. So our chromatin was our big spaghetti-like mess inside our DNA, or inside our nucleus, all our DNA. And they shrink down to chromosomes so that they kind of look like X's. So our nuclear envelope breaks down, and then our spindle fibers appear. When we think of prophase, I like to think that, or remember that pro means before. So since it's before, it means it's our first step. And then the important part about prophase 1 is we have this crossover occurs, and then giving us genetic variation. So what happens is we have chromosomes that are similar line up next to each other. And this is what is called homologous chromosomes are aligning. Homologous, we've learned that word before, just means the same. So similar chromosomes get next to each other, and then they get kind of tangled up. So this is what's called crossover. But what happens is, as they're tangled up, some of our genes switch. So we can see the genes that started in the blue end up on the red, and the genes that started on the red end up in the blue. So this is how we can get some changing of genes, some changing of DNA, and this is how we can have siblings from the same parents look very different. It's because of this crossover, because it never crosses over the same way more than once, or very rarely does it, I should say. So for example, my brother and I, we look very different. We have very different body types, skin color, eye color, and it's because the crossover between our parents' cellular division in meiosis was different. So then we have metaphase 1. It's the same, just like metaphase. So our spindle fibers appear. We can see these little spindle fibers appearing. Our chromosomes line up in the middle. Remember, metaphase, middle. Then we have anaphase, we have anaphase 1, which is just like anaphase. Our chromosomes are pulled apart. I like to think of this like a fishing pole. They reel in their line, which are our spindle fibers, get reeled in towards our centromeres. And I like to remember ana means away, so they're getting pulled away from each other. Then we have telophase, telophase 1, which is just like telophase. Our chromosomes grow back into chromatin, and we start to get our spindle fibers to break down, and our nuclear envelope starts to appear. So we can see these little kind of whiter parts making a circle. That's going to be our nuclear envelope, whereas our chromosomes are starting to look more and more like spaghetti, which is its chromatin. Then we have cytokinesis 1, which is like cytokinesis normally in mitosis. It's where our cell divides into 2 separate cells. We mentioned before that it's a little different for plant and animal cells because our plants have a cell wall and our animal cells do not. And then we have prophase II. This is just like prophase, but now we have 2 cells to do it towards, but each of them are going to do it at the same time, but in their own cell. So chromatin, spaghetti-like stuff, shrinks to chromosomes, which look like X's. Our nuclear envelope breaks down, and our spindle fibers appear. Then we have metaphase II, which is just like metaphase, but with 2 cells. So again, they're going to go through it at the same time. Meta, we think of middle, so our chromosomes line up in the middle, and we also have spindle fibers attached to the centromere, which is the middle of our X, and again, meta, middle. We have Anaphase II, just like Anaphase, but with 2 cells. Our chromosomes are pulled apart. This is our fishing pole reeling in our line. Ana, away. So they're getting pulled away. Telophase again, Telophase II, just like telophase, but with 2 cells. Chromosomes grow into chromatin. We can start to see nuclear envelopes appear, and our spindle fibers break down. And then cytokinesis 2, just like cytokinesis. Cytoplasm split into 2 cells, and it divides. And again, different for plant and animal cells. So in one big picture, the summary of this is we don't have our interphase, so we have interphase out here, and then we have prophase 1, metaphase, prophase 1, metaphase 1, anaphase 1, telophase, and cytokinesis 1. Then we have prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. And that all leaves us with 4 new cells, 4 different cells from the beginning. So now that we've kind of gone through it, this is a short video showing the animation of it all, that I just wanted you guys to watch real quick and maybe try to see if you can picture each one of these different phases happening in our animation. So I know that was a little quick. I hope it makes sense though. It is much like mitosis, very, very similar, just with really prophase 1 being an important crossover step. We got to remember that crossover occurs in prophase 1. Other than that, everything is pretty much identical, pretty much the same to mitosis. We just do it twice. That's why we have to label it metaphase 1 or metaphase 2. But I hope that makes sense. We'll do some more practice with this going forward, but if you guys have any questions, as always, please let me know.